Stem Cells Across the Curriculum is a collection of modules and educational tools designed to make stem cell research tangible, relevant, and responsive to social justice. Central to the curriculum is this radial infographic that traces the development of stem cell research and its associated technologies by integrating the biological, ethical, social, and legal dimensions across space and time. There are many different sources for human stem cells, and much of the controversy associated with this area of research centers on the nature of the source. Scientists have developed technologies in the lab, or in vitro, to isolate and manipulate human stem cells, and this radial infographic serves as a visual narrative depicting how each human stem cell type was identified and sourced. Towards the bottom left corner, we see how stem cells work in the body, or in the in vivo environment. Stem cells contribute to human development from the moment of conception, through embryogenesis, and even in adulthood. Within the human body, stem cells reside in niches where they can be triggered by environmental cues to differentiate into the specialized cells of tissues and organs, and regenerate, creating a pool of stem cells that can be harnessed later in life in response to aging or injury. It is important to recognize that this panel in the infographic depicts human development in vivo, or in the living body. How cells behave in the body differs from how they behave in vitro, or outside the body and in a petri dish. This distinction of studying cells in the body or outside of it in a lab environment is an important one in stem cell research, and thus we have created a visual separation of the two to make this even more clear. The ability to study cells in a petri dish in a lab was accomplished in 1951 with the establishment of the first human cell line, otherwise known as HeLa. This groundbreaking work on cancer cells allowed scientists to explore the technology of tissue culture, an essential tool for cell research. Tissue culture contributed to the development of in vitro fertilization techniques and the emergence of an IVF industry that results in the surplus or extranumerary human embryos that can be used as a source of embryonic stem cells. The radial nature of the graphic reflects human ontology. As you move clockwise, you see that sources of stem cells can not only be found in the embryo, but also in the fetus and specific tissues in the adult. Furthermore, genetic reprogramming can push adult cells back to their embryonic state, as in the case of induced pluripotent stem cells. Moving outward along each spoke of the radial, one can identify the origin of the stem cell, view a visual narrative of the manipulations needed to create a stem cell line from this source, and understand the derived cell line's relative differentiation potential as it relates to basic science and regenerative medicine. Cells that are pluripotent have the most potential to develop into any cell of the body, while those that are multipotent are more limited in their cell fate potential, which may give clinicians more control in a therapeutic context. A rollover allows the viewer to zoom in on the characteristics and details of each spoke while maintaining the ability to compare the stem cell line with others in the radial graphic. For instance, the first 3 embryo sources require both egg and sperm, while the next 3 embryo sources only require eggs due to genetic modifications in the lab. Likewise, the genetic constitution of each cell line is also highlighted to emphasize important DNA reprogramming events necessary for reprogramming cells to pluripotency. In the downloadable PDF of this infographic, the outer concentric circles contain hyperlinks that take the user to the research paper announcing the identification of the stem cell type and to articles and video clips that illustrate the broad range of stakeholders concerned with the ethical, social, and legal dimensions associated with the development and use of this particular stem cell line. The spokes are also represented in downloadable Zoom graphics, which measure 11 by 17 inches. These Zoom graphics serve as mini encyclopedic digest, providing the learner with a deeper understanding of the development of this cell line and its potential uses. In the future, each Zoom graphic will also be available as an animated PowerPoint slideshow. The radial begins with the first human embryonic stem cells created in 1998. In Jamie Thompson's lab at the University of Wisconsin, he isolated these human embryonic stem cells from embryos that were created in excess during IVF. In that same year, primordial germ cells were isolated from fetal tissue, removed from medically terminated pregnancies by John Gerhardt's lab. Because these two sources of human stem cells are ethically contentious given the moral status of the early embryo, researchers turned to new biotechnologies that could support isolation of human embryonic stem cells for study, such as clones, parthenotes, and cybrids, and also began to explore adult stem cell sources, representing the various stem cell lineages such as endoderm, mesoderm, ectoderm, and germ. The radial orientation of the infographic is intentionally designed to bring the user back to the human body. Though stem cells are removed from the body and manipulated in vitro, this research expands our basic scientific knowledge of stem cell behavior in the body and can also lead to the development of therapies that ultimately will be used in the body. Lastly, to provide a more contextualized view of the human dimensions of stem cell research, we have mapped case study modules to the infographic to illustrate how race, class, gender, ability, and religious pluralism intersect with policies and practices associated with this field. The first case, HeLa cells and HPV genes, immortality and cancer, interrogates the relationship between cancer and stem cells and the role of human subjects in research by reviewing the events that led to the development of the first human cell lines. HeLa, isolated from a cervical cancer biopsy of an African-American woman named Henrietta Lacks, and WI38, isolated from fetal lung tissue collected from a therapeutic abortion. The second case, oocytes and blood, gifts and commodities, reviews funding initiatives to specifically diversify the supply of stem cells used in research and therapy, and uses a feminist framework to analyze the ways that tissues of the body, such as fat, oocytes, menstrual blood, cord blood, bone marrow, and ovaries, are being reframed in a capitalist market. The third case, Disease and Disability, Hope and Hype, looks at stem cell research in the context of cure, using a disability justice lens. In this module, learners explore the ethical issues surrounding the removal, sale, and transplantation of human adult stem cells, the emerging markets for cell therapies, and regulation of safety and efficacy of such therapies, as well as the increased allocation of public resources for medical approaches to public health during a time when funding for social services that support health is being reduced. The fourth module, Stem Cells and Policy, Values and Religion, reviews the changing political and legal landscape within the context of differing value systems, and how these differences shape the emergence of new professional societies, oversight committees, guidelines, laws, and bans at the local, national, and international level. Each case study is a teaching and learning module with associated teaching notes, essential learning resources, and student assignments. The case modules can be used individually or in combination. As seen in the case studies area of the website, each case module reviews specific biological concepts and principles related to cell division, cell differentiation, genomic reprogramming, and cellular aging, as well as the related biotechnologies needed to research stem cells such as recombinant DNA and cloning techniques, in vitro fertilization and genetic diagnosis protocols, microarray analysis, and emerging cell typing and imaging analysis. This curriculum affords learners with the opportunity to engage with a dynamic and exciting field that can support scientific innovation while maintaining socially just practices. The ways in which institutions and committees regulate or oversee human stem cell research reflects the importance of procedural justice in terms of who can participate in this field as a scientist, policymaker, or human research subject, while the policies and guidelines that emerge from these regulatory and advisory bodies influence distributive justice in terms of who has access to the knowledge and products of this emerging field. We invite you to explore the philosophy of our approach in the who and what section of this website, and resources available in the curriculum and cases, and the infographics and media portions of this website, and we welcome your feedback.